So what are some of the evidence of anti-aging benefits from stem cell therapy? So there are some studies I want to show you which are really interesting. So this is general background information. Age does lead to progressive deterioration of the cholinergic and dopaminergic systems. So that's in both our muscles and our brain. Muscle mass is important. Of course, maintaining brain capacity is important as we age. So the concentration of neurotropins in the brain and muscles decline, leading to reduced neurogenesis, accelerated muscular atrophy, and that can lead to impairment both cognitively and physically. Aging can lead to exhaustion of stem cell population, as we've seen in the statistics. And various stem cells can exert neural protective effects and enhance functional recovery by secreting neurotropic factors. And the concentration of this T-bars is byproduct of lipid peroxidation. So just kind of waste product that increases significantly in the brain, muscle, heart, liver, and lungs of aging rats, and along with a decreased blood vessel density. So these are just signs of aging. And what they did, well, first of all, they have seen that adipose-derived MSCs can increase the concentration of neurotransmitter acetylcholine and also some growth factors, brain-derived neurotropic factor and nerve growth factor, and those can improve cognitive and physical function of aging mice. And adipose-derived MSCs can also secrete very high concentration of VEGF, so helping with blood formation and extending lifespan in an ALS mouse model. And even these muscle-derived progenitor cells can extend health span and lifespan of progeria mice. So these are prematurely aging mice. They do this by restoring microvessels and muscle fibers by secreting different factors. So in this experiment, which is very interesting, they started at age 10, which is kind of middle age for mice, rats. So these rats, they give them either control or amniotic membrane-derived MSCs or adipose-derived MSCs. And they give these cells intravenously through the vein in the tail once a month throughout the rest of their lives. And the stem cell group showed improved cognitive and physical function of these naturally aging rats, lifespan extended by between 23 to 31%. And by the time these rats, right, they were 10 months old, by the time they reached 20 months of age, in the control group, only 30% of rats are still alive. But in the ones that got stem cells, there's between 70 to 100% of them, the rats are still alive. So there's a big difference. And by 23 months, all the rats in the control group have died. But between 60 to 72% of the stem cell group are still alive. And some of the transplant cells were they found them to differentiate even into neurons. So there's a significant differentiation rate, so which is fascinating. And when they looked at the different factors that we talked about, so there's brain neurotropic factors, they were restored to a level higher than when they were in younger rats. And muscular neurotropic factors were also restored back to levels younger than the younger rats. And they looked at the blood vessels, and their density also have increased, comparable to the level of the young rats. So the restored angiogenesis. And the tissue injury was almost fully restored. And the monthly transplantation of these stem cells have markedly increased the rats' stamina and cognitive function. So that's, we're talking about vitality, right? So cognitively and physically. And then acetylcholine concentration in both the cerebral spinal fluid and the muscles have been restored back to the younger state. And the gene expression which changes, right? Genes start to degrade as we get older, and those changes were also reversed. So they were able to measure all these different, tangible outcomes. And the number of the host stem cells in aged rats is a lot less, right? It's only 27.8% of the younger rats. But when we infuse these new stem cells, they increase the number of these host stem cells to the level that's comparable to younger time. And the expression of certain things that suppresses neurogenesis, so this particular molecule, that has been fully reversed. So then you can restore neurogenesis. And stem cell therapy, so in conclusion, can restore cholinergic and dopaminergic nervous system and microvessels and muscle mass and antioxidative capabilities. And then I want to show you another study, which is really interesting because this study started a little later. So they started these rats, these are mice, at age 15 months. So this is human equivalent age of 75. So these are much older mice, and 50% of their peers have died. That's how they determine the time. And they were giving them intravenous bone marrow transplantation from bone marrow cells of the younger mice. So they gave them also through IV route 6 times within 3 months, so every 2 weeks. And the mice that's only 3 to 15 weeks were the donors. And the result is that the maximum lifespan of the transplanted mice were increased by over 30%. So that has been pretty consistent. This extending of lifespan by 30% has been consistent. And the survival time from the beginning of the experiment was increased by 3.2%, a fold, which means that, instead of having 1 month left, if you got the stem cells, you could have, 3 months left, right? So at age 19.3 months, it's kind of funny, but the last mouse of the control group died sedentary, almost immobile, hunchbacked with poor hair. The transplanted mice were active, had even spine and shiny, even hair. So they're vibrant. It's not just living longer, they're living well. And these observed lifespan definitely, was accompanied by, the healthy life period. And there's chimerism, which means that one individual have 2 sets of genetic material. So there's these new cells did stay in the mice, and a lot of them did stay. The chimerism was 28%. So this is very encouraging for us as humans. And this is just a study on humans. It's not a big study, but we still need more human studies. This is 20 patients with chronic inflammation. They were just getting 1 IV infusion of umbilical cord blood-derived stem cells for anti-aging purposes. So half of them showed improvement in CBC, so in their blood count, their chemistry comprehensive, panel, their liver function, and their cholesterol. And they evaluated the changes for anti-aging benefits, at 24 hours, 2 weeks, and 3 months, and showed improvement in skin, hair, nail growth, energy level, libido, mood, sleep, and pain levels. Yeah. So just the last study, I'm just going to show a little about frailty. So frailty is actually a big problem. When we age, people get very frail, and that's correlated with hospitalization, institutionalization, disability, and death. So there's huge percentage. Well, there's 7 to 12% of adults over age 65 fit the definition of frailty, but there's pre-frailty, which means they fit partial criteria, is as large as 35 to 50%. So there's a lot of frailty which is associated with comorbidities, with all your organ systems, and neurological and psychiatric issues. So, and they show that there's a strong link between frailty, inflammation, and the impaired ability to repair injuries to the fact that there's not enough stem cells. And aging induces a quiescent to senescent switch. So you may have stem cells that are quiescent, right? They're just, hibernating. They're not doing anything. They're waiting. And then aging made them switch to senescent. So before they even can get to work, they're already aged, they're old, and they're causing problems. And they have shown that MSCs can undergo senescence. So as we go older, they can become older and become less effective, and their capabilities gradually disappear. And intravenously delivered allogeneic MSCs, so from somebody else, are safe and can produce significant improvement in physical performance and as well as inflammation markers.